presentation is this engineered bioremediation of LNAPL polluted soil water. So I am going to use very simple words. Okay, technically very simple words so that everybody can get what I am going to say. So here the keywords are the engineered bioremediation. I think everybody know about the engineer. I will talk about the LNAPL and then I will come to the bioremediation. And then polluted soil water sources. Everybody knows that we are having natural resources either in form of the and water pollution. So looking at this title of this conference, I am going to talk about the soil and water sources only. So when somebody says LNAPLs, what does it mean? So actually these are the research area in which I am working. So already Sandeep has introduced, so I can go to skip that now. So now look at this keyword LNAPL. LNAPLs are the hydrocarbon oils. LNAPLs are the hydrocarbon oils which are mainly derived from the petrochemical industries. Whatever the petrol or petroleum derived oil which we see at gasoline station, at different automobile workshop or any chemical industries where they are using all these oils. These are the hydrocarbon oils. The property, the LNAPL stands for non-aqueous phase liquids. Why it is called as non-aqueous? Because if you are going to put hydrocarbon oil along with the water, it is not going to mix, right? We'll see the separate phase of the oil and separate phase of the water. So oil, hydrocarbon oils and water are not miscible. They are immiscible, and that is why we call them non-aqueous phase liquids. It doesn't mean that they are not going to solute in the water. These are in the separate phase, but a small quantity of these hydrocarbon oils are get dissolved into the water. If you fill a bottle which is containing maybe some petrol and fill it with the water, then you can smell the water having the petrol smell, right? So it's very small quantity of these hydrocarbon oils get dissolved into our water sources, and that is enough for degrading the quality of the groundwater or the surface water resource. Most of the remaining hydrocarbon oils remain in a separate phase, and that is why it is non-aqueous phase. Now, these hydrocarbon oils can be categorized in two types based on their density. If the density of these hydrocarbon oils are lighter than water, we call them light non-aqueous phase liquids, designated by LNAPL. If their density is higher than water, we call them DNAPLs. So NAPLs are categorized as light and dense non-aqueous phase liquid depending on the density. Now what happens when these hydrocarbon oils are get released from the surface? So this is the ground surface or below the surface. So there are different sources of these hydrocarbon oils. Mostly like whatever the storage tank which we are having. You go to any gasoline station, there is subsurface tanks containing all these hydrocarbon oils. There are many other storage tanks and pipelines which are supplying these hydrocarbon oils. So there is a probability of leakage of these hydrocarbon oils from these tanks or the pipelines, or there may be some accidental spills at the top of the ground. So this is the source for the release of these hydrocarbon oils. And initially these tanks are made leak proof, but with degradation in the time, there may be some tectonic impact or maybe the age of the material by which these tanks are made of. So there are leakage, and these leaks are going to be quite unpredictable, and one cannot quantify the leak from these tanks. So after releasing from these sources, these NAPLs move vertically into the soil. I am talking about the top horizons of the subsurface of the soil, and this is moving because of the gravitational force. It is similar like if you take a glass of the water, you pour it over the surface of the soil, the water will go down. Okay, some will go in the lateral direction also, but primarily this oil, which is another fluid, is going to move vertically in this unsaturated zone. This is a unsaturated zone where the soil particles are not fully filled by the water. There is an adjoining area which is denoted by yellow color, and this is the soil air, because these contaminants are volatile in nature. So if you leave them into the open atmosphere, they call them pollutants. Now a portion of these in this direction, a small layer of these LNAPLs are made over the water table. So maybe 5 liter leakage of the LNAPL can cover a large area over the water table, and a very thin layer of the LNAPLs can be made in the capillary range that is nearby to the groundwater table. I'll give one example. Heathrow Airport, if you go there, there is 6 to 7 meter depth of this LNAPL standing below the airport. Where this one coming? This is coming from the leakages, accidental spills, which is taking place in the refilling of all these aircraft in the buses. So it means they will start moving in the direction of the groundwater flow. But their vertical movement is very limited because they cannot penetrate into the saturated zone because their density is lighter to water. Now, when the groundwater is moving in this direction, a small component will also get dissolved with the water, and that will also pollute the groundwater sources. And when at the down gradient receptor, if somebody is going to drill a well, but after reaching to the groundwater table, they will keep moving downward direction because the density of DNAPL is higher than water. Now they will encounter some impervious barrier, something like that. Here you can say fine grained clay liner here, clay lines here. So they will be retained here. If the sufficient amount is there, they will start it going down further until they reach a rock strata in the deep groundwater sources, and then there they can reside happily for thousands of the years. The life of the DNAPLs is more as compared to the LNAPLs. DNAPLs are more persistent as compared to LNAPLs. Why? Because in the deep saturated zone, there is a limitation of the oxygen. In this zone, we can see some transfer of the oxygen from the ground surface to the soil to the water table, where these LNAPLs are there, and they can be naturally attenuated. Naturally, they can be degraded. But in case of DNAPLs, they goes into a deep strata where the oxygen is very limiting, and if the oxygen is limiting, the things becomes anaerobic, and anaerobic degradation of these hydrocarbon soils is quite slow as compared to aerobic attenuation. And the examples of the LNAPLs are like monoaromatic hydrocarbon, as I told you, like petrol, or BTEX compounds. Everybody has heard these BTEX, like benzene, toluene, xylene, ethylene. So this toluene or benzene, these are mostly used as a solvent. You go to any pharmaceutical industry or any other chemical industries, mostly toluene is used as a solvent. So this is the prime source for the contaminating form of LNAPL. All the chlorinated solvent, like trichloroethylene or TC, these comes under DNAPL category. DNAPLs are more carcinogenic than LNAPLs. And the major source of the DNAPLs comes from like with the dry cleaning industries. All the dry cleaners, they are using this PC and TC. Also the automobile industry, the automobile workshop, where they are using for cleaning the parts of the motorbikes and the other motor parts. So these are the prime source of the contaminants. Fine. So this is the, now question comes that once these LNAPLs and DNAPLs are released over the surface or below the surface, they are going to pollute our natural resources, and natural resources in form of the soil and groundwater, as well as air also, but we are not talking about the air. That once these natural resources are get polluted from these NAPLs, whether it is LNAPL or DNAPLs, how to clean those sites? So there are many methods, there are many remediation techniques, treatment techniques which are used for decontaminating such type of the polluted resources. So here these are the, mostly these are applied at a very limited extent. Where the contaminated natural resources are at very limited area, one can apply the physical treatment techniques. If the large water sample from the field, there are number of the microorganisms which are present in that sample. Indeed, we cannot see these microorganisms from our naked eye. Okay, these are very small size bacteria, microorganisms or microbiota which we cannot see. But always at every site, whether it is polluted or it is clean site, one can expect the number of the natural microbes. So bioremediation is a grouping of technologies which uses natural microbiota to degrade or transpose the pollutants. These pollutants could be organic or inorganic type. Just now we are talking about the LNAPL, DNAPL, which are organic type of the pollutants, which could be located into the soil water system to less or non-toxic daughter products of byproducts. So in bioremediation, natural microorganisms are utilized for eating the contaminant which is there in our natural resources. Now these natural microbiota is mainly comprising the bacteria, fungi and the yeast, but the number of the bacteria is more as compared to the fungi and the yeast, and that is why when somebody says microbiota, it means implicitly we are talking about the bacteria. Now the main problem with this bioremediation method is that this is very slow treatment technique. One has to wait for cleaning a polluted soil water sources maybe for 10 years. Okay? So the major drawback of bioremediation is the time framework which is required for treating the polluted natural resources. Now question comes that how can we increase this natural rate of bioremediation so that we can treat the polluted resources within a stipulated time. And for that one has to talk about the enhanced bioremediation, or one can say it is going to be engineered bioremediation. Right? Now this engineered or the enhanced bioremediation can be in form of biostimulation, bioaugmentation, or phytoremediation, that is called as plant enhanced bioremediation. What happens in biostimulation? So let's say we are having a natural resources which is free from any contaminant. You go to that site, put some pollutant there at that site, the site will be contaminated. But naturally there are some microbes are there, as I told you. These microbes will become active and they will start eating the pollutants as a carbon source. For microbes, whatever the LNAPL, DNAPL or the hydrocarbon oils are there, it is much like a carbon source for them. So these natural microbes will start eating or removing these contaminants from the site. Okay? Now that requirement for the treating is going to be more time consuming, and in biostimulation what will happen, more supplements are provided to the microbes. Supplements means nutrients are supplied to the natural microorganisms in form of the nitrate, in form of the other fertilizers like sulfate and all. So if you provide more food to the same number of the microbes, they will be more active, more healthy, and they will work their work more efficiently, right? The cleanup process is going to be faster. So that is known as biostimulation. In biostimulation, nutrients or the oxygen, sometimes oxygen is also supplied because oxygen is an electron acceptor. So when we provide the air into a polluted site, that will increase the availability of the oxygen, and when the oxygen is more, microbes are going to be more active, and that is going to be increase the ultimately the rate of bioremediation. So that is the biostimulation. Another way of increasing the rate of bioremediation is the bioaugmentation. In bioaugmentation, extra microbes are introduced at the polluted site. You go to the market, purchase some microbes, go to the polluted site and introduce these microbes at the site. What will happen? The number of microbes are going to be more, which are going to be responsible for degrading the contaminant. It means if the number of these microbes are going to be more, we can expect the less time is going to be taken for removing the contaminants. Fine? So that is known as bio-augmentation. So certain natural microbial strain or a genetically modified variant is introduced to treat the contaminated soil water site. So that is, now in literature review there is a very much confusion. Some scientists, they advocate that biostimulation is much a better technique than bioaugmentation or vice versa. So there is ambiguity that which technique is superior than the other. So in that case, one has to see the environmental conditions or the prevailing site-specific conditions for applying either biostimulation, bioaugmentation, or integrated technique of biostimulation and biostimulation. That is going to be all these together. And then third technique, that is the plant enhanced bioremediation. So assessment of the engineered bioremediation techniques, the role of the site-specific environmental factors are becoming very crucial. Whenever you see that this is a polluted site, now question comes whether we go for the biostimulation, bioaugmentation, or bioremediation, or a combination of these two or more than one techniques. One has to consider the site prevailing environmental, whether it is arid and semi-arid condition, whether the soil moisture content is more, whether the availability of the water is high here, what is the temperature of the site, all these, what type of the soil is there, what is the pH value of the water which is going to be polluted. All these environmental factors which are over the surface and below the surface has to be taken into account for taking a decision for applying biostimulation, bioaugmentation or bioremediation. The second key word is the integrated impact of the environmental factors on the freight and transport of these networks. Sometimes we see the impact of the temperature. There are many environmental factors which are going to affect indeed bioremediation. For example, temperature, soil moisture content, pH, as I told you. So when you say the temperature is high, we can expect the metabolic exchange of the microbes is going to be increases with respect to the time till a certain value. Okay. So these environmental factors has been assessed individually. One guy has assessed the impact of temperature on the microbiota, another person has seen the impact of the pH on the microbiota and bioremediation, and the third person has seen the impact of the oxygen on the microbiota. But no systematic study has been conducted to see the integrated impact of all these environmental conditions together on bioremediation. So that is another for topic which can be looked for when somebody goes for the engineered bioremediation. Now question comes that how the plants can help us in increasing the rate of the bioremediation. So this figure shows the different levels of the plants which is going to affect positively the rate of bioremediation. It's going to be nutrients bioavailability. This is a plant, and mostly plant roots which is there below the surface is going to release some carbon source which are known as root exudates. Root exudates are the carbon source which is the most portable food for the microbes. So plant roots releases some substrate, and that is going to be more as a carbon source for the microorganism which is present in the polluted site. Also there is a direct uptake of the contaminant. If the pollutants are there, let us say in this root zone, water is taken up by these plants through these roots. It is transported to the root biomass or the above ground portion. So when the water is going to move from the root to the shoot biomass, the contaminant which is dissolved in the water is also going to be taken up by the plants. So physically the contaminants are also removed from the water uptake process of the plants. Also the oxygen transfer rate is also increased. You might have seen when there is a vegetated site, the availability of the oxygen is going to be more, because oxygen can penetrate through the heterogeneity which is created by the root parts, and also the oxygen is released from the root cells at the night. So that is going to also catalyze or you can say increase the rate of bioremediation. have been instructed to wind up the things within 5 minutes. Alright? So this is the process that how the plants are going to utilize this bioremediation rate in different ways, which I told you that the contaminants are taken up by them, the oxygen availability is increased, the root exudates are released into the root zone. So all these integratively going to affect the bioremediation rate positively, and we can clean up the polluted natural resources more faster rate. And these are the subcategories of the phytoremediation, which depends on the type of the contaminant which we are going to target, whether it is heavy metals or whether it is going to be organic type of the contaminant, and what type of the polluted resources we are talking, whether it is soil or simply water, polluted water or a soil water system. So depending on the target contaminant and the target natural resources, one can categorize the phytoremediation in different subcategories, which I am not going to explain now. Next. Okay, now what we have done, we have for getting this answer of biostimulation and bioaugmentation, we planned a series of practical experiments in the laboratory, as well as numerical experiments, mathematical model. From a very small scale, that is a batch scale, completely mixed system, to a pot scale or mesocosm, which is denoted here, to a large continuous column setup. So at very small scale to the intermediate scale to a large scale, we have investigated this biostimulation and bioaugmentation using practical as well as the mathematical model experiments. Next. Medicine. What you are seeing, we have taken the small batch experiment, completely mixed batch experiments. And what we have done, we have put these batches at two temperatures, which can mimic the day and night temperature of the site. For example, daytime we can see the temperature is very high now, maybe 30 degrees Celsius or 25 degrees Celsius. At night it is going to be 15 degrees Celsius. Next day it is going to be daytime. So there is a diurnal fluctuation in the temperature, and which has not been incorporated, which has not been investigated by the researchers. So in that case we are seeing different temperature variation by putting the microorganisms at two different temperatures to show it as a like winter scenario or maybe the summer season, depending on the temperature which has been selected in form. Now these are the graph, no need to get panic. I am just going to tell the simple things here. The solid line shows that there is no natural attenuation of the contaminant. Okay. So this is the concentration of the pollutants on y-axis. On x-axis it is the time of formation. So with respect to time, there is no attenuation. The contaminant concentration remains almost constant in all these three batches. So these batches kept at constant temperature of 10 degree, 21 degree and 10 degrees Celsius. What we have seen, when we increase the 10 degrees Celsius temperature, the rate of removal time, the time of removal is get reduced by 1.5. Okay. Or in other way, we can say by increasing the 10 degrees Celsius of the temperature, the rate of biodegradation increases by 2. So that is the impact of the temperature by taking them as a constant temperature. But in reality, temperature doesn't remain constant daily. It is fluctuating day and night. So we have seen the fluctuating impact over here, and what we have seen, when the microbes are facing such type of the temperature stress, their degradation capabilities get diminished. So efforts should be made to minimize this temperature variation. So we collected the wastewater from our campus and the groundwater, which is wastewater which is coming from the household in 1:1 ratio. It means nutrients has been supplied from the wastewater. So these are not the, actually this is not a wastewater, but it is a naturally, the nutrient rich resources. So nutrients are coming from here and the polluted groundwater coming from here. So that is going to be a type of the biostimulation. Biostimulation will provide the extra supplements so that the microbes can work more efficiently. In third case and the fourth case, we have seen the impact of the plants. How the plants are going to favorably increase the rate of attenuation. In third case, we simply bypass this last water through a wetland, frequent wetland, small frequent layers in these branches. In the second case, but we have not found any difference between natural attenuation and the forest. What could be the reason? We were expecting, because we have hypothesized that when we are providing the nutrients to the microbes, they will get more food and they will work more efficiently. The metabolic actions are going to be more faster. But in that case, there was no positive role of these extra nutrients. The reason was that later we found that whatever the groundwater which we have collected from the site was already enriched with the nutrients. So before applying any treatment technique to a particular site, one has to investigate the status of the soil water sources. Okay. What are the different environmental factors? What is the nutrient status of the polluted resource? And definitely in the third and the fourth case, we found positive roles of the plants, and the attenuation rate is faster in the third and the fourth case. And here we see the role of plant next. This is the modeling work that this is a paper which has been published from this work. So you can compare this next. And then we have integrated this column, large soil column experiment with the treatment wetland. So this is going to mimic a weather zone. In reality what happens, we are having a vegetated site, some plants or some vegetation is there, and then we are introducing some contaminants in form of maybe with the fertigation or some accidental species, and these contaminants will leach out through the root zone. So to mimic that zone, what we have done, we filled this column with the soil, and we connected this column with the desired wetland, treatment wetland. So polluted water was coming from this stock solution here, and the water flowing from this treatment wetland supplied at the top of this column, and it is vertically moving downward direction here to show that how this polluted water moves into the and then we are seeing this how these contaminants are moving. These are the different cookers which has been taken the sample. The dotted dots show this analyzed samples, and the solid line shows the numerically models experiments. And then we have upscaled it at multi-dimensional level using a 2-dimensional tank setup as well as the 3-dimensional tank setup to see the things which happens in the field. And this is the work which has been done to see the impact of the plant and how the plants can accept the heavy metal that is going to be lead. In that case, we have investigated at the university as well as the UNESCO actually by taking the wetland plant. This is the wetland plant which is very common in the Europe, and we investigated that is going to be quite positive in taking absorbing this contaminant heavy metal and transporting the heavy metal from the root to the shoot bypass. And this is the one of the beautiful example where we applied this technique. This is the site of the Gandhavan. Vatsa and Gandhavan, many of you have seen. These are related to the Lord Krishna, where a lot of natural ponds were there in ancient time. But at present time, if you go, most of these natural ponds has been enclosed by the anthropogenic activities. Mostly whatever the landfill, they have made it like a landfill, and they have made some construction work also around it. So there is a thermocone which was like this when we started this project, along with the NGO that is known as Brush Foundation, the daily based NGO. But that NGO has divided this one to this stage, and then they filled the water in this cone. And what we observed that after a week or so, the water was very smelly. The water which was filled in this cone was very fresh initially, but after a week or 10 days, the foul was coming from the odor was coming from this water. And that was the wet molecule of the water is turning from within a week. So to get rid of from this water quality, to improve the water quality of this pond, what we propose to make a treatment wetland around this pond. In this picture you can see here more. So this is the treatment wetland. So groundwater is passing from the surrounding area, it is passing through the root zone of this plant, and also whatever the water which is filled in this pond is interacting with the plant roots here, and the water quality is improved very significantly, along with the some sprinkler system which is provided here. The purpose of the sprinkler system is not for the aesthetic reason, but to provide the extra oxygen. Oxygen, if the dissolved oxygen is going to be more, we can expect the natural attenuation of bioremediation is going to be more faster. The role of plant, along with the nutrient acceptor in form of the oxygen, was able to improve the water quality in this compound, and now it is looking like that. I think with this example, I wanted to give an international example also, which we applied this technique in the Saudi Arabia also for decontaminating the contaminated soil water sources, where most of these petroleum products are derived. But I've been in such a take only 5 minutes.